Hey guys, welcome to another class. Today we'll be talking about plant nutrition. So before we talk about plant nutrition, let us define what nutrition itself is. Now, nutrition can be defined as the process by which living organisms obtain and utilize food materials from the external environment for metabolic processes such as respiration, growth, excretion, and reproduction. So nutrition is just simply the process whereby living organisms get their food materials from the external environment. Now they use food that is gotten from the external environment for metabolic activities such as respiration, growth, excretion, and reproduction. Now let's define plant nutrition. Now plant nutrition basically is photosynthetic nutrition or holophytic nutrition. Now, photosynthetic nutrition or photosynthesis can be defined as the process whereby green plants manufacture their own organic food from simple inorganic substances such as carbon dioxide, water, sunlight, and chlorophyll, and produces oxygen as the byproduct. So let's break that down. In photosynthesis, plant manufactures their own food. Plant makes their own organic food from simple inorganic substances. And the inorganic substances are carbon dioxide, water, sunlight, and chlorophyll. And they produce oxygen as the byproduct. Now, photosynthesis occurs in two stages, which are namely the light reaction or the light stage reaction and the dark stage reaction. Now the first stage, which is the light stage reaction or the light reaction, is dependent on light, as the name implies. Now the light energy is absorbed by chlorophyll. So we want to talk about the processes now. So what happens is light energy is absorbed by the chlorophyll, which is used to break water into hydroxyl and hydrogen ions. The chemical formula of water is H2O. That is two atoms of hydrogen and two atoms of hydrogen and one atom of oxygen. So what happens during photosynthesis in the light stage reaction is that chlorophyll absorbs light energy, and that light energy is used to break the water into OH and H plus, OH minus and H plus. So that is one hydrogen goes with one oxygen and another hydrogen is separate. Now this process, the process of splitting this water molecule by light energy and hydroxyl and hydrogen ions, by light energy into hydroxyl and hydrogen ions, is called photolysis. So this process of separating the water into hydrogen and hydroxyl ions is known as photolysis. Now this stage is very important to transfer the light energy to chemical energy. The chemical energy is ATP. And the meaning of ATP is adenosine triphosphate. Now, this stage also makes reduced NADP. And the meaning of NADP is nicotinamide adenine dinucleotide phosphate. Now, in the dark, this energy is converted to chemical energy in organic compound. So that is that for the light stage reaction of photosynthesis. So what happens in the dark stage reaction, which is the second stage of photosynthesis, is that sugars are built from hydrogen and carbohydrates through a series of complex processes involving enzymes. So what happens is that in the dark reaction, carbon dioxide, which is CO2, combines with a 5-carbon sugar. The 5-carbon sugar is ribulose diphosphate to give two molecules of phosphoglyceric acid, which is PGA. Now this phosphoglyceric acid is converted to carbohydrates in the presence of ATP and NADPH2. Now other products of this are proteins and fats, which are made according to the needs of plants. So those are the other products of photosynthesis, proteins and the fats, and they are made according to the needs of the plant. Now, the actual main product during photosynthesis, the food which the plant is actually making, is simple sugar. Now, this simple sugar is formed is partly used by the plant. Now, excess of it is then converted to starch immediately for storage. Now, this starch is then transported to the other parts of the plant through the phloem tissue for storage. So it transports that starch into other parts of the plant for storage, and this process is called translocation. Now, let's talk about the importance of photosynthesis in plants. One is that photosynthesis is used for the production of food for living things. Photosynthesis is used for the production of food for living things. So since plants are the producers, and all other organisms directly or indirectly get their food from plants, so photosynthesis is just the process by which food is provided for living things. Another one is that the process photosynthesis purifies the atmosphere by making use of the carbon dioxide in the air. Humans breathe in oxygen and breathe out carbon dioxide or carbon dioxide, that is CO2. So photosynthesis helps to purify the atmosphere by taking that carbon dioxide, which is not useful to humans, then releasing oxygen. So it's purifying the air, and the oxygen which is useful to animals and humans. So it releases oxygen into the atmosphere. So that way, photosynthesis helps to maintain the oxygen balance of the atmosphere. So photosynthesis also serves as a building block for other substances to be formed, e.g. protein and oil. So photosynthesis serves as a building block for other substances to be formed. So we want to describe an experiment to test for photosynthesis. Now, in one of our previous videos, we've described or we've mentioned the steps to writing down an experiment. So we're going to be describing an experiment with those steps, starting with the aim. Now, what is the aim? The aim is to test for the presence of starch in the leaf. Now, you also list out the materials required. The materials required for this test or this experiment include fresh leaves from an outdoor plant, then a beaker, boiling tubes, a dropping tube, and a white tile, and iodine solution. So those are the materials required for this test. Then the method, now the process of this experiment, which will be written under method. The first one is to boil the leaf in water for 4 to 6 minutes. Now the reasons we boil this leaf in water: one is to kill the cell. Two is to make the enzyme inactive, to inactivate the enzyme. And another one is to burst the starch grain present. So that's why we boil the leaf, to kill the cell, to inactivate the enzyme, and to burst the starch grain present. So the next step is to dip the leaf into a test tube. And the test tube will be containing about 90% alcohol. Then the purpose of doing this is to decolorize the leaf. Then after this, the leaf is dipped into water, and the purpose of doing this is to soften the leaf. Then after that, you pour a solution of iodine on the leaf. Then this leaf is kept in the dark cupboard, Then a fresh leaf is plucked to test. So the purpose of doing this is to test for starch. So we pluck a fresh leaf and keep that separately. Now, the leaf that is freshly, the observation now, what is going to be written in observation? The leaf that is plucked from the potted plant, a new plant. So when we are pouring the iodine solution is poured on both leaves. The leaves that you had poured the alcohol on and the iodine solution. Then a leaf that you didn't put in the alcohol solution, but we put iodine solution on that. So what is observed is that the leaf that is plucked from outside turned blue-black. The one that we plucked from outside turned blue-black with iodine solution, while the other leaf remains colorless. Now what happens is, what will now come as a conclusion is that the leaf, since the leaf in the real experiment has turned blue-black with iodine solution, that shows that photosynthesis has taken place, or you can say that shows that starch is present in the leaf. So with that, you've completed the experiment. Thanks for joining us today. With that, we've come to the end of today's topic on plant nutrition. Next lesson, I'll see you again. We'll talk about another topic in biology. See you next time.